Okay, good morning. This is a lecture number 7. We are going to discuss classification problem. And this is a field which very successful in machine learning field. Okay, a lot of applications, and for example, the neural network was developed for classification purpose. By the way, we do have a formal course ID. Okay, so if anyone, if you recommend anyone to take the course next semester, to let them know this 675 is the formal ID for this course. Okay, so we are going to discuss several approaches in classification, and this is a topic which is very important for machine learning, but less important for single cell data analysis. I send you a link, lead you to the Twitter. One of the investigators just complained yesterday about their study and to classify the cell type. Yeah, I can show you this later. But anyway, for make this course complete, I have to mention all this machine learning approach used for classification purpose. So we will discuss neural network. We will discuss KNN, K nearest neighbor, logistic regression, LDA, naive Bayesian, and see how can we evaluate the successful of classification, whether you design an approach. And the tool you can validate by using your own data, but once other people use it, give random results, that's not a good indication of your classification approach. At the end, we will discuss some of this cell type identification, classification problem, and use it in the single cell data. Let's first go to, I want to do something different this semester. I want to go to the last page. Maybe, yeah, let me show you the Twitter thread. I realize some of you may not have Twitter account, and so I created this thread. Basically, it's a copy all the post under this thread and make it like a web page or a blog page. So you have this link, you can read it through. And I don't know Simona, she's from which institute, but she complained about this most cell type assignment method will fail, and for totally novel cell type. Of course, they will fall because most of this cell type assignment is based on the known marker genes and assigned to cell types. If you have a new cell type, you have to define by yourself, and that's why the biologists had to get involved in some of these key rare cell type which nobody studied. For example, we have collaborators study cyberblast. So he is the only one in the world discovered, defined this cell type. So he will never be able to use any annotation tool to find his cell type because they're not in the database. He's the only one in the world have that particular cell type defined and studied. So this is I hope you can find time to read this thread. Okay, I will jump to what we have in this tool, and remember. So I will load this and okay. Assume we haven't done this cell type identification. Remember, we can do automatic cell type identification, and you tried this, and this labels is pop up and gave each individual cluster a name. We can also do it manually. And for example, we can select by brush this. And there's a small brush next to this automatic annotation to say, okay, I want to use manual annotation to brush. Oh, sorry. Let me, yeah. I know what to do. Okay, so you know this, right? You can automatically annotate the cell type if you have a class. We have done clustering already, and this will assign for each cluster an ID of cell type. You can also do it manually and by select, for example, this cluster, and then you click this. This gave you indication that this is one-time analysis. You will not record any assignment to the data. You just have your personal impression of this. Say if this mouse data, you select the database from mouse. Okay, so this time it gave you the suggestion that this should be endothelium cell. Okay, so you can do this again by fibroblast. Okay. So I want to explain what's behind this action. What's behind what we are doing here. Okay. So before even we go to the formal introduction of all the clustering algorithms, at the end of this lecture, I probably will ask you what this method, the algorithm belongs to which category of that. Is a neural network? Is a Bayesian method? Is it supervised or not? So let's see. To the side, I'm going to jump to the very end of this slide. Jump to the end. Because I leave this algorithm at the end of this page. Okay, the algorithm I used for that interface is defined, introduced in this paper. Okay, so this paper is a web server to provide a marker gene database and use that database to help you to assign the cell type. This database is one of the best. We used it extensively. However, because of funding issue and this database maintenance has been discontinued. It's a Sweden, I don't know which university, Uppsala University. It's from Sweden. They stopped updating. So the marker gene database has been frozen and it was stopped updating for a while, but still we can go to the website and download a database. Okay, I show you how to do it. Okay, so cell type marker, that's where we can get this database. Okay, for this cell type for human and mouse, you have 5 genes as marker genes for this particular cell type. Okay. Size. And you can download the data. Okay, this is too big for me. Okay, so anyway, you have a database for marker genes. Let's see. This database, you have cell type. One cell type. Okay, and with each cell type, you have gene marker genes, but I'm writing this one. Gene 1, gene 2, maybe that will show the cell type 1. And then you have a gene 3, 4, 5. Data definition for cell type 2. Okay, so that's the database. Marker gene. Okay, no problem, right? So you don't have equal numbers of the marker genes. Some cell type well defined, you have a lot. Some have only a few, right? And maybe some genes are shared, and gene 1, gene 4, gene 6. Define cell type. That's 3, right? That's possible. You have this list. So in this side, we will have the cluster. What? Last term. Okay, you have the cells. This cluster may have 50 cells, another maybe have 150. What we want to compute, when I click one of this button here, I will compute, for example, I select this cluster 1. The group I select, I will compute a number for this cluster belongs to cell type 1. This cluster belongs to cell type 2. I will compute a score across all the cell types in the database. If this database contains 140 major human cell types, I will compute for cluster 1, 140 ID numbers, scalar numbers. If this number is large, I will give you the feedback and say, okay, the output for this assignment for type 2. Cluster 1 is cell type 2. Make sense? Okay. So if I do manually, I can select another group cluster and repeat this, and this cluster may give me cell type 3. Okay. If I click that automatic button, then this procedure will automatically run and make each cluster cell type. Easy, right? Make sense? Oh, okay. So now the problem we reduce to how can we compute the score here, right? How can I compute the score that indicate class 1 belongs to cell type 1? Or what's inside here that we should use? What information each of these? What should be in there?
Cells that we send that those genes specifically.
Yes.
The group.
Yes. So if you want to see, okay, this class one belongs to cell type 3, you have to have these 3 genes on average expression level is higher. And remove of this. So what we will do is to compute the average expression of all the marker genes in them. And then I do a summation. So I think Z G1 as an expression, average expression of G1 gene in class 1, right? I will do a summation G4 plus G which 6. I will check the average expression of the cell in this group. Okay. If our average is 3 genes, expression level, average expression level in this group of cells is high, this value should be large. Okay. So another we have to consider is the number of genes. It's not fair for this particular cell type because it's only 2 marker genes. And in that case, we have to do the average. You have to divide this by 3. So you have 3 markers to take into account. There is only 2 here. Is that easy?
Yeah.
Okay, so that's just principle. Just that easy. I just have to do this very quick. Check the gene here, average here, and sum the value at the end and pop up the lines, right? That's it. Okay. Well, in this formal definition, and you do have a Z here, okay, which is this Z, average expression of particular marker genes. Okay, I is marker genes. And also you count how many marker genes. If you have 3, this big N, 3. Okay. They take this qubit square root, the sum of transformation. So this way to make it more justified. So this big sigma is this plus all the marker genes in this for this cell type, right? And there's a W here. What's this for? This to give each gene a weight. So this, for example, this one kind of weight of 1 is our weight 4. What this means is if you have a marker genes and gene 1 updated twice in some gene may be very popular and present in all the cell types. So you have to give a weight that by down. If there's particular genes in gene 99 only present in cell type number 4, cell type 4. So this is a very unique contribution for your almost 100% definition for cell type 4. So you have to weight this higher. Okay, so this weight, this W just give you some either weighted up or down by the frequency of your complementary presence in the dictionary in this database. Okay, so this is the gene Y you have to weight by its gene weight, which comes from here. 4, 6. If you put this together, you understand what they're doing. So what I did, I just follow the paper and implement it, and it works. By the way, this equation on the top here is to compute how to compute the weight. The weight for the compute. And give the value from 1 to 2. And is this a gene present in all the cell type? We have a group 1. If the unique gene, we make 2. Other type of the frequency is in the middle. You can define this by yourself if you have a function defined that's slightly different. Okay. All right, any questions? I want to keep your mind fresh when it's early morning and you'll still be able to focus. Yeah.
If we have a cell type which is being defined, which have the 2 marker genes
Yeah.
of 2 different cell types, at that time should we consider the p-value?
Yeah, you can assign a p-value actually. Yes, there. Even for this algorithm, you can assign a p-value here.
You said like the cell type 3 has the 3 marker genes, right? These 3. So supposed to be this cell type is not defined properly. At that time, which gene should I consider as a marker for this type of cell type? Should I consider the p-value of G1, G4, and G6 at that time?
G1, G4, G6. I don't think you have any p-values for individual genes. You can see, well, I have a p-values for this particular score. So high, so unlikely to be some high. So there's some kind of p-value you can compute. But for the purpose of what we have here is the power up with the largest value compared to other cell types. I don't care how confident or specific this. So I didn't put a p-value for those.
So in short, we can consider the p-value when we are defining the marker genes for the cell type.
Yes. Yeah. So for each of this, you can compute the p-value. Okay. So if you believe this 0.01, 0.005, and they both considered to be significant, I can return both to let you select. So it could be this, could be that. But I don't think there are any formal statistics to take care of this. So I didn't implement it, but if you look at this paper, I covered up this. So next is p-value can be computed with the Fisher exact test. You'll see this paper, the last sentence, a p-value can be computed. Is computed using one-sided Fisher exact test. So it's possible to do this. Okay. Okay, let's go back to our slide number 1. So this is a comparison between classification versus clustering. And clustering, you just don't care. You don't need the training data, and then you don't have a label for the data points. You just group them. Well, classification require you put a label onto this clusters or individual data points. So in machine learning, this is a two-step process. You have to train your data first to make you a predictor first. And this predictor is a classifier and to cluster new data points, okay? So that's why this is a typical supervised learning, supervised algorithm. And the clustering is an unsupervised algorithm. So that's easy to understand. You have to have a labeled training data, either green data points, red. And to learn this, the structure of your data, the distance, and how you separate them. And then you build this algorithm with a machine, okay? And then deployed for the applications, then you have new data comes in this classifier, just put, okay, new data, red, green, to give you the results. As I said, the machine learning field uses extensively to recognize the objects. The picture is apple, it's tomato, it's cow, and their facial recognition. Okay, it's you or not. The algorithms gave you output, either a labeled output to different clusters, and all gave you yes or no answer. It's you or not. So this is a like image. You have to put this image as a data and to give to the prediction function and give you output. So a lot of effort has been devoted to how can you convert an image into a data point, data, okay? Because the computer will take a vector, so that you have vector, the column by column, and but the image is 2D, or you have a color, it's 3D. How can you convert that into a vector? That's a lot of so-called image feature learning, and to let you to learn the majority difference or similarity between each pictures, to learn the most characteristic features and give to training. And then you learn the model will be used to test your new object. There are many different ways to construct a classifiers. The logistic regression, Bayesian classifier, perceptron. Perceptron is the unit of neuron, which we will use in the neural network. I will show you how to do it. We will not discuss, maybe next week, to discuss the supervised support vector machine decision tree. Okay. So today we are going to focus on this column, left side of the column. Okay, perceptron, and this is a unit of neural network, and it simulates the neurons. And when biologists work out, okay, how the neurons work, there's a lot of input, and then generate the signals, and there's output. So computer scientists just simulate this action of this. So let's see how this works. I will give an example and use some numbers to indicate, to let you know, okay, how exactly a single neuron will can be used as a classifier to classify the given data sets into different categories. And once you know this, you have multiple neurons, you can connect them as a network. That's a neural network. But this one is the simplest one, and it's useful. So you have input. Input is your data. We know the data is a vector, okay? Column vector. You will, so for example, this have m dimension. So you have, if you have 2 dimension, you have two data. And if you have 3 dimension, you have 3 numbers in this column vector. Where you have m different dimension, then you will assign a weight for each variable in the dimension, and then summarize them and process by the neuron. There's a function to tell you is which class this output is supposed to be. So we will, I'm going to use a simplest model. We have a data set. We have a data point, for example, there are 4 dimensions, so that will be a vector with 4 numbers. So I'm going to use a neural network, a neuron to take this and to give me a binary output, say either positive or negative. Okay, so I have an example like this. Okay, my first data is. Say, one. So I'm going to give this to my neuron. And this has not been defined in all the weights. One, two, three. So this weight maybe as a random number on the all zeros. Okay. So I want the output to be -1, 91. Okay, so what I will do is to have this data to V by the times W, this weight parameter. So these four numbers give you the setup for this particular neuron. And this setup will not work because this I will do a dot product. Okay, I will have a V dot W, we can transpose and put them together. And this will give 0, so this setup definitely not work. So I have to change the parameters here. If I change this to 1 minus 1, 0, 0. I don't know if this works. No, I think the answer for this product of these two will be positive, greater than 1. Okay, so this is not a good. This is not a good set. So I will have to try until found set like. In my example, this 1.40, 0.7. So this will work. So for this input data, this one will be a negative value. So here I just have a sum function. If you have the dot product between this given input data and this weight is positive value, then give you a positive 1 or otherwise negative 1. Okay. In this case, I want for this particular data input data, I want negative. And then this one. So this is acceptable setup for this neuron. It has been trained. Okay. However, our classification is cannot just use one data point. I want to try another, so I gave to another one which is 0, 1, and 5. Something like that. Yeah. So this is my training data number 1. I want training data number 2 also work. I try to get this to be negative. And you can do the dot product again. And that's negative. So my training set is okay. My setup of the parameter is fine. And I can give this V3 another training data. I want to have, for example, this example minus +1, 0.5. I want the output for this to be positive. You can give it a try and also positive. Now my training finished because I use 3 data in my training, and then my output is what I want. Negative, negative, positive. this parameter set up is fine. So I'm done. So I finish the training of this single neuron and let it to perform, behave as I want it to behave. I want it to behave. If I have this data, it will be +1, this will be -1, and this will be -1. This particular setup is fine. Okay, in real training, we don't have to do this one by one. I can do this V vector. We have V vector containing 3 big points dot vector W train and give me so this will be +1 and -1. And just do once, so that's very quick. One computation. What I have to do is try to optimize the values here. There are 4 values. Here I have to try and then until reach to the result I want. This is just one solution. If I have many different kind of solutions for this small example data. Right? So that's a neural. That's the basic of this neural network. Any questions?
Major aim or task of this neural network is to find the W.
Yeah. That's called train your model, train your neural network to find a lot of W. Some deep neural network have millions of these parameters. It's updated by once you give the training data again and again, and it's do a small adjustment and change until you get the output. Okay, so that's an example. Let's just draw it on here, and you have 3 input data, and then you can put them as a matrix, like W, and then you can set up this parameter combinations. If you have a parameter set like a set number 1, and it will not work because you want the result as a negative, negative, positive, but this parameter set number 2 will satisfy what you want to achieve. Okay, so when you set the parameter to the parameter set number 2, you finish the training of your neural. You are ready to go for picking the new data to test. Okay. So for this particular perceptron, there's only one operation, just weight all the input and sum it. So you can achieve this very simply by just do a dot product, a matrix multiplication. And oftentimes you want to have a further process. So in our case, the further process is just a sum function. If I have a positive value, give you a +1. If a negative value, give you a -1. So in that case, is this activation function is just a sum function. However, for some of this, you can make this function more complicated. Okay, you can have a say threshold after the finish crosses threshold, it gives you +1 or -1 or some other functions in activation function. But this is the summation for the input signal and weighted and take some. That's the same. Okay, then you change this. So that's the structure of the neuron. You can have different activation function selections, and you can use this logistic hyperbolic. Those are the commonly used ones. Okay. sigmoid function in take this form, and the very commonly used in used as a activation function. So the feature of these functions is that your x-axis is from a negative infinity to a positive infinity, and their turning point in the middle here, and then you can set up parameters to make this either narrow or wider, and so you have a threshold which in the continuous range to give you, for particular range you have this output. It's between 0 and 1. So that's the feature of this function. So when you do the logistic regression, you have all the input that you use as a sigmoid function to help you to decide a soft cutoff, basically. So you have a range, and in this range you have either a probability between 0 and 1, and other functions like linear and not to give you this feature. Okay, it's hard cut or monotonic increment. So this is called a hard decision. The no decision, you just increment your probability by the increment of x values. Well, this gives you a soft cutoff, soft decision, once you have 80% of this, 20% of that. So again, this is like a 2D illustration. You have weight 1 and 2. This is the interception, and then you do this. Basically, that's dot product. You transform this weight matrix to allow the data matrix X, and then to do the multiplication of the matrices. You get the separation either +1 or -1. So this source code to help you understanding how you guess this values. Okay, you don't want to just use a random number generator to generate this test. That will be slow. That's the brute force and you try out. And this way is do an iteration and update the weight through a couple of iterations, and then you achieve the difference between your previous setup of the parameter and the next round. No much change. When your norm is smaller than certain cutoff, you will stop updating your vector set, your parameter set. So this is the Python version. This is MATLAB version. You have also the access to Julia version. So they're common use. Okay. Now we put multiple neurons into a network. So I think in this graph, I'm not so sure whether they gave this input data or this all orange or green point dots are the neurons. I think they're neurons. So you have two layers of neurons. In this case, your output will be as an input to give the next level of the neurons. Okay, so you have some input in front. Okay, to compute the output, like our +1 or -1. Or if you logistic regression curve, you have a value and between 1, 0, and you gave them the output of this as input to all the next level of the neurons. And you copy into 4 identical copies and gave them as input for each of the next level, because the next level have 4 neurons. You make 4 copies and give to the next level. Same thing, you have the second neuron and get output and give them to the 4. So every next level of the green neurons have 3 input from the first 3 neural layers, and then synthesize and give to next level. Okay, next level can expand the number of neurons or decrease number of neurons. You can have either enlarged space of the parameter, allow it to have a much more refined space to digest the input information, or maybe you want to compress your data into a small lower dimensions to ready to be output for something, some purpose. Yeah.
Understand where it comes from, the numbers 1, 2, 3, 4.
Yeah. So 1, 2, 3, 4 is from a, for example, that's your input, that's your output. In our case, is 1, 0, probably the negative one, but if you use other functions, noise, then you can have values say from 0 to 0.5. Okay. But in this case, it is 4 to 0.5. And input other in another level. In this case, this of course neuron will accept the data itself. When you go deeper, it's between neural connections. It's the output of the first neuron and the next level of exactly depends on how many neurons will survive. So in this case, you can build whatever levels of neurons you have, and each level you can have different numbers of neurons. Okay. But at the end of this very complex situation, you can just do the matrix multiplication. Okay, you have the ability to do it.
And those weights are the ones you calculate.
Yeah, calculate from the previous steps. I think that's the chop in the middle of somewhere, and then you have first layer 3 and the second 4 neurons. And in between the computation is matrix multiplication. So that's why you remember the improved algorithm to accelerate matrix multiplication a little bit. 1% if you have a huge impact in the industry, because this operation has been done so intensively in all this neural network computation. So here is some examples. This is a single layer. So you have input, then you gave to the single layer of neurons and gave output. This is like two layers, we call them multi-layers, where you can make many more. And I don't know the definition, maybe after 20, you call it a deep neural network. Deep means you have more layers. If you have a structure like this, you have a input, have a lot of neurons, and then in the middle, there are hidden layers, it's very low dimension. And then you use the low dimension, and that populates the information again to expand it to multiple neurons. So this structure have a name, so the encoder decoder. We use this to first we will compare the input and with output to make them as similar as possible. That means the information has been compressed and expanded, and you don't lost any information, because if you have an out layer and input and output layer have almost identical information, then that means your data can be compressed into very low dimension in the middle. Okay, people use this a lot to do the compression of the information. So you use neural network to compress the information and then expand and compare your input with output, and then change the parameter in the middle, make sure they as similar as possible. When you reach that, the setup of your parameter in the middle will help you to compress your information, and you use only the middle layers will be useful for you to learn your original data. Okay, that means this encoder decoder keeps the information identical, but only use very low dimension of the representation in the middle. Make sense? Okay. Yongjian actually use this structure to learn a data set, single cell. Then he can compress information of all the input gene expression matrix with low dimension and put the cells or genes in a 2D space to look at the relationship. And then he delete one genes, and then delete a gene and in the input, and he try to use the original decoder encoder structure to see, okay, what's output looks like. The output will be different from the if you use non-delete input. So he compare the output and to see, okay, what the impact of this deletion of the gene. If we have time, I will ask Yongjian to present his work. He use encoder decoder to learn the matrix of the single cell and to infer the virtual knockout, so knock out the gene from the input, and what let the encoder decoder structure to reconstruct the output and see if you don't knock it out, what will happen. If you knock it out, what will happen? You can compare the output to infer what will happen after you knock out the gene. Make sense? Yeah.
So that way you can predict the mutations.
We can predict if you knock out a gene, what other genes will be impacted. In theory. Okay, as I said, so this is not related to what we are doing in the single cell, but I just want to know how we can convert this image file into the input, the vector data, so allow the neural network to learn. Okay, so this is called a convolutional neural network. Basically, you have pictures. How are you going to extract the information from the picture? This is a 2D or 3D, depends on your definition. This how can you learn these positions, the features? So basically, you are doing to do a quick scan. You use a small window as a source pixel and scan the picture from the top to end, and then you learn from that to learn a new matrix. So this process is called convolution. It allows to extract information and normalize it and put in this new matrix as your input data. You see this like a sliding window, scan your image, and then extract the information, summarize, and put in the pixel. Then you move your sliding window to next step and do another. Then you synthesize all this convolutional feature map and try to give to the neural network to learn, depends on your label, is positive or negative or cat or dog. I will play this YouTube illustration. I don't think there's sound here. Hmm. I don't know what to do. We can take a break and in this 40 time we can. Maybe the person. Okay, let's use this video to illustrate how this convolutionary feature map is working. Feature mapping. So you have image like have a capital A on there, and the pixels indicate the image and the different colors. And then you use a small window. This is like 3 by 3 to scan the pictures from each pixel and then summarize information and project to another layer. Basically, learn this. There are different kinds of this sliding window design, whether you will consider the boundary or are you going to randomly jump or so. Next slide, I will show you these options.
How do we know that which information we have to take from the input and convert that to the output?
Oh, your input, you have to be standardized. Say if you have 182, 82, 182 by, so you have a matrix of image. So you have the resolution, the fixed. So you start from the pixel 1 and take this 9 pixels and start the scan. In this case, the video maybe showed you another levels of scan. You finish one and then scan that again and to extract information until it becomes some feature map. Then for a new image, you just scan that again and to match the feature map and say, okay, it's most likely to be A. Then C, something like that. Okay, I cannot play this directly in the slide, but what I did is Google search this author's name, and then in the YouTube channel, it can easily be found. So I will I should put this linked here. So I put the link here. So okay, so this page is from a GitHub. The authors created those image files and animations to to show you different kinds of this sliding window scan design and whether you consider their boundary effects or not. Okay, there are different ways to do it. Anyway, so machine learning, deep learning, they're just a word, and they're deep learning probably means you use this neural network contains a lot of layers. I don't have a definition of how many layers you have become a deep neural network. Okay, next. Next is a very simple method, which is called k-nearest neighbor. Yeah.
I'm not recording.
Recording. I think I still keep this recordings on. Yeah, I'm recording this. I didn't pause during the break, so it should be fine. The only problem is I don't have the picture for this blackboard, but this slide you have to have that. So this we will first look at the. So K means you define let's see, you consider how many K numbers of nearest neighbor as your connected partners. For example, if you consider K as a 3, then for this new data point, this star, the red star, then you have 3 neighbors to be considered, and 2 of them are purple. So by vote, and this star should be purple class. Well, if you consider the neighborhood contain 6 nearest neighbor, then you have to consider this big circle. Within this big circle, small local network, and you connect with 6 members. There are 4 belong to yellow category, and the 2 are purple. And in that case, this KNN, when K equal to 6, and this dot belong to yellow class. Okay, just look at your local neighbors. And then you do this by vote. Is this a good method? It works okay, especially if you have a very complex shape of the data. It just looks like a local and what your neighbor is. And this method, of course, depends on the K value you selected, and also depends on the distance you compute between data points. So different way you compute your tested data point with others will probably have different results. Okay, so we mentioned there are some different ways to compute the distance. Of course, Euclidean distance is most commonly used.
You should place the star in the middle.
No, start is your test data. It can be anywhere. So you have your labeled data and you know where they are. Okay. Now, for example, you have 2 cell types and you know there, and then you introduce a new cell type in your t-SNE embedding. Okay, this new cell type right in the middle of 2 clouds, and then you just look at a neighborhood and you belong to this most likely. Okay. So in this case, you don't consider the real distance. You just consider, okay, that's in this radius, in this circle there, how many. You can also consider the real distance. So if you have distance from this data point to here, it's far away, and then you want to weight it down a little bit. And then if you're close enough, you have a cutoff say, okay, it's close enough, it gave you a very high weight. So you can somehow define this weight to give you the close neighbors. And not only this two, but also consider the distance between the neighbors. So you weight your distance. Logistic regression, as I said, it's easy, and you have your computer statistics that you say, okay, first you fit your data, you compute. So those values, for example, in this case, the values is between 1.5 and 3.5. So you have to define a function and where the separation should be and what is the cutoff supposed to be. And then you define the shape of this curve. Okay, so all the parameters you can change. It depends on the training data. Okay, the yellow and the blue are different categories of the two classes. And they have a different distribution in their values. And then you set up this curve and finish your training. So once you have new data points come in, and then you can draw a line to the probability. Okay. And you can draw a line and found its y-axis values, and then that probability belongs to either blue or yellow class. Okay, next is called linear discriminant analysis, LDA. LDA is pretty much like PCA. Okay, if you don't look at those data points, if you don't consider them as a two categories, you just put like black dots, then you will learn a PCA and found, okay, this could be the first PC because it's help you to separate the data points overall. But in this case, our purpose is not to display all the data points. Our purpose is to find a rotation to rotate the data and let them project onto this PC and then make them separable. So what we can do is to learn a PC, which in this case, we should learn a PC like this. Okay. Rotate your data points and allow them to separate as much as possible. What you will optimize, you will achieve is the center of this red dots from the center of this blue dots are far as large as possible. You maximize the separation of the data points. So the PC will select will be this one. Okay, the separation is most will compare to other kind of rotation. Make sense? So this is different from a PCA because your input is not the data point itself. It also contains of your data points. Okay, so that you want to achieve not the only separation of the data into maximize their dispersion, but it's to separate two cluster of cells, two group of cells, data points labeled differently. Okay, so our objective can be summarized using this equation. you have the mean. Okay, so now we reduce the dimension to 1, this 2D dimension. 2D example. So you have your mean for green ones is here, and you have your mean for red data points here. So you want to maximize the difference between them. So that's why you use one of the mean minus another, take a square. Okay, so either big or small, you square, that means the difference is big. So you want to maximize this and you want to minimize their within group variation. Okay, this variance, that's for the green variance, green S squared, is variance for green data points, and this red one is for red one. So you put this summation of this total within group variation in the denominator here, and you want to maximize the whole value here, which separation between the two groups mean, and minimize within group, the summation of within group variation. And that's the solution. So basically, this again is the rotation of your data points by multiplied by a rotation matrix until you achieve this maximization of this value. Okay. So this example showed I can't see the small templates here. So this small example indicates the solution is not this one. It's that one here. The reason is, although you see this average difference between this separation is large here compared to this, but because there's variation in the denominator here, so the variation is very small here. So you make actually this is the best solution. You can separate two groups if you look at your data from this angle. Okay. Okay, so this is algorithm for LDA. We compute mean vectors. Okay, so this mean for each class. Then you compute this total mean. Okay, you consider multiple classes. And you compute within class variation. Okay, so if you know how to compute the covariance matrix of the variation, okay, within group all the data points. And then you compute it between class. So now you consider the data points from one class to another to compute this variance. And then this eigen decomposition algorithm here, and to get the projection you want. Okay. When you compute the mean here, the purpose of compute this mean is to help you to compute the variance, the matrix. Okay, so you take an inversion of the within class variation matrix and multiply it by this between class, and then take eigen decomposition. You get eigen vectors, which correspond to the top eigenvalues. Those vectors are the solution. So this is the MATLAB code for LDA. Just that simple. So here you have labels, the different class, different groups, and you compute this within and between class variation matrix. And then you do this eigen decomposition. And then you order them, and then the solution is this, and then separate them. There are YouTube channel and the YouTube video to explain this in more detail. Again, that's the difference between PCA and the LDA. LDA deal with labeled data. PCA is non-supervised, unsupervised. So LDA, you need to have, LDA is non-supervised, but you need to provide labeled data so it knows what to separate. Any questions? Okay, naive Bayesian method, it's not too hard. It's based on this Bayesian theorem. So let's consider an example. So now, if I gave you a data point, see what this data point with this question mark belong to, either green one or yellow one. So without any further information, I will first to count. There are 10 green data points and 7 yellow points in this 17 total. So if I know nothing about this new data point, it's most likely to be green, because the probability in the population, the frequency of green dots is higher. So this value is called a prior knowledge or prior probability of the class. So we have this first, and then we will look at to compute the posterior probability. So we have a P green, which belongs to green, is it's prior probability. And then look at this small circle. If you have, let's see, the probability of sample. So you sample around it like for let's see. Yeah, now you consider just this like a neighborhood. Within this neighborhood, if you have you have this how to see this, okay, if you are green in this neighbor around the test data point, you're supposed to have 3 out of 10 being green. Okay, so this is like a sample related local probability. and then your prior probability is global. And within this circle, and you supposed to be green is that 3 out of 10. It's hard to get head around this. So you multiplicate these two probability, you get the posterior probability. That's your solution of whether the posterior probability, whether this belong to green category or yellow category. Okay. Another illustration is to use this keyword, whether if you have a keyword, this keywords in your email, whether it's a spam email or not. So that you have this training data to help you first to establish those marginal probabilities and prior probability. Then you have your test, and then you have your keywords in there. And you compute the posterior probability whether this message is a spam or not. I should have a link to follow up this. Yeah, there are summaries for the readings if you like to go deeper. Well, now how can you evaluate your method is good or not? Those are the commonly used. So like cross-validation, people call the CV, is an internal test you should do by yourself. And then you can use precision or recall, that's the fraction of the corrected or sensitivity, or you can use this called ROC curve, okay, to evaluate the method developed for the classification purpose. So I will explain what the CV is. So when you do finish your classifiers, you can separate your data or input data into 2 random sets, into 10 random sets if you do this 10-fold CV. For example, in the round 1, you separate your input data, use 90% of your data as a training, and then 10% of your data as a validation. So you are going to use this blue data sets, blue fraction of this data set to train your model and get the parameter. And then you test, use this 10% of the test validation data sets to test your data. Because when you do the training, you didn't give the information of your validation data set. So this can be used as an independent test. So you can get your accuracy of your classifiers. So, okay, your round 1 validation get accuracy 93%. Then you can reshuffle your input data and then give another 90% as a training data and the train your model, change your parameter, and then use rest of the 10% of the data set to do the test. In this case, you only achieve 90% of accuracy. You do this 10 times. Then you take average. So this average is your final report accuracy of your algorithm of your test. You can see, or you can claim I use a 10-fold CV method to get my final accuracy of my algorithm to be 93%. Okay. Get the idea? So this is an acceptable validation method. When you do the publication for your classifier method, you have to define this golden standard. You know the data set, you know the correct classification. And then you use 90% training and 10% test. Do that 10 times, you report tenfold CV accuracy. People will accept, okay, your accuracy is really robust. Okay. Not just only test once. Okay. I think you know this confusion matrix. In this corner, you have a true positive and a true negative, and you will try to avoid false positive, false negative in these two corners, upside and down, bottom. So those are the formulas to compute accuracy or recall false positive rate, sensitivity, specificity. It's very easy to make it wrong because even some of this Wikipedia make mistakes sometimes. Okay, one of the technique you probably will encounter when you look at those algorithm evaluations, you want to oftentimes you get the ROC curve. Anyone use ROC curve? No, no one. Okay, so this is 2D curve, and in your y-axis, the true positive rate. Okay, so if you have a good classifiers, you want to have true positive rate as high as possible, right? So you have 80%, 90%. You want to have your method to be in that corner. And in here is your false positive rate. Well, let's see. So you want to make this lower. So you want your method to be on your top left corner as much as possible. If you have a method located here, means you have 90% true positive rate and less than 10% false positive rate. False positive rate is also the 1 minus specificity. So if you put a specificity here, and your values will be flipped, and you always see a ROC curve. Actually, you have a true positive rate with a sensitivity on your y-axis. Let me just draw this. And the specificity on your x-axis. We always see these. Specificity. Okay, so sensitivity is higher the better, okay? 0 and 1. Well, specificity is from is better to this 0 point. So 1 is better for this and 0. Yeah. You see the values flipped. Okay, in order to guarantee you have best message. That's in this column. So you have to look at this carefully. If you say, okay, false positive rate here, that's fine. So you have 0 to 1. But oftentimes the specificity in that case, 0, 1 have to be flipped in order to achieve for your left up corner to be the best results. If you have a random method, you should have in here in this model. Anything above this is the pattern. Okay. The curve is created by multiple data points, multiple points. So how can you, for one algorithm, have multiple points here? It's because separation, the definition of your parameter. Sometimes you achieve this, sometimes you achieve this. So on average, you need to have a curve which is towards up, left, corner. That means your method is good. So this curve is defined by multiple of data. Each point is corresponding to one of the setup of your algorithm. So your algorithm will be defined by this. Make sense? You can achieve very good sensitivity and specificity in this up left corner. And sometimes your parameter in your higher sensitivity specificity not good enough. Sometimes you achieve this when you have enough parameter space for your model, for your algorithm, create a curve. So your curve is compared to another competitor's curve. Their curve would be this, and your curve is better than that. Okay, so you are better than that. People consider the area under the curve. The area under the curve, so the right algorithm is better than the area under the curve for the green algorithm. So you just compare the size of the area. This is called the ROC curve, okay? Let's see some of these single cell type classification tools. These two examples is to assign cell type based on nearest neighbor using similarity measurement. This method uses Spearman, Pearson correlation, and this use cosine similarity. You know how to compute the cosine similarity, cosine dot matrix, the vector multiplication. If you cannot remember, I suggest you look at the previous week's slide. There's a cosine similarity compute. Is that by dot of AB divided by the norm of multiplication of norm? There are supervised learning algorithm. For example, this one use SVM, and this one Surety use random forest. I'm not so sure here, and the Surety's package are very famous. There are many algorithms have been developed, and there are evaluation papers to compare, say which one is good, which one is bad. I think I have some of this evaluation paper link if you want to look at them, and they compare so many different methods. Okay, some of this just LDA directly, some of the random forest, some SVM, KNN directly. So they use this like baseline classifiers. So very naive method, not for single cell per se, but you can use them, give it a try as long as you have the labeled data and train your data already, train your LDA. Okay, you have to rotate, you know how to rotate your data points using a training data first. Okay, then you have new data points, new cells coming into prediction. predict. The problem with this evaluation method is they probably use very small data set and will define most of the T cell, B cell. And it's very easy to separate because they need to have a golden standard in order to compare different evaluation methods. But those methods, those situation, relatively simple. You don't have to use any algorithm. You can separate them very easily. Well, when the real problem comes, just like in this Twitter thread, the investigators just discussed, none of this will work at all. So this pipeline explains this particular method. It's supervised method for using of training data, different cell type, this matrix, then SVD, that's a PCA basically. And then the features, I don't know. I don't suggest you spend time on this. Okay. Then you start to predict, do the same thing for the predict the model. This is another single cell net enable quantified. Let's look at its pipeline. You have training data and already labeled cell types. It even includes cell type from a different species. I don't know what it is. And then build your classifier, and then you have a query data. Transform, feature selection, random forest. So basically, this is called a random forest prediction classifiers. Then the claim you can have this heat map for global view identity. Then Berlin plot attribute. Well, I don't know. This is another paper I selected, this pathway based. It can do classification, clustering, construct interaction networks. Yeah. Sounds very powerful. I don't know. A lot of this real application, people just use manual annotation and they select markers by their own definition. They just not mention any classifier algorithms. I think that's the approach we should take. Yep. So we already discussed this. That's what I used in my package. So now you have a better understanding when you click that, what happened in FixPlot. So now the question for you is, which category of this algorithm belong to? It's not a neural network, definitely. I don't have an answer. I don't know which group of this classification algorithm this method belongs to. It's search a database, just look at a dictionary, some kind of, then use some score to give the values for each cluster of cells, different cell type assignment. I don't know what this algorithm belongs to, which category of this classification tools. Another idea I want you to think about it, can you do this algorithm for every single cell instead of a cluster of cells? Because when you do the cluster of cells, you take average of all the genes in that 500 cells, whatever. Can you do this for every individual cell? If I give you just one cell, can you do this? In principle, you should be able to do it. You can assign for every individual cell a cell type. What's the problem for that?
Size which are within a fast growth method. I mean, you get the marker from differentially expressed genes, so what if it finds different markers for two cells which have been defined around the same last. Because it's, I don't just look for the different genes, differentiated genes.
No, the method of not using the,
Otherwise, if it just uses the most highly expressed gene in each cell.
Yeah.
It is to take into account the other genes that are also expressed. In that algorithm.
Mhm. I don't understand. I don't know.
I have seen algorithms which take the individual cells.
Yeah.
As
Assign cell type for every individual cell.
but then I map them back onto a Disney or they are usually a mess.
Oh,
So I don't. I have tried doing this.
oh, I think what you said is if you predict a cluster of cell, for example, they belong to fibroblasts as a group, but if you look at individual cells, maybe some belong to this cell type, that, but as a whole, as a majority should be fibroblasts. We can give it a try, maybe.
I tried it.
We can give it a try. Yeah. Okay, I think that's all. Yeah. Let me know if you have any questions. Yeah.
So, the class said that some things have more weight than the others, because they have to appear more.
No, when they appeared less. If you have a common gene which has a marker, yeah, they should not be that important.
What if you have the same number of mini genes?
Same number.
Yes, you have